At high enough doses, ionizing radiation is a universal sterilizer. It shreds the delicate double helix structure of DNA, making cellular division biologically impossible. For decades, the damaged nuclear reactor at Chernobyl stood as a total biological vacuum, a location completely hostile to living organisms. But deep inside the reactor's most highly radioactive bunkers, scientists found something unexpected. Thick, black biological growth was creeping up the walls. An environment designed to permanently extinguish life was instead serving as a catalyst for a thriving subterranean ecosystem. This growth is driven by three specific types of fungi: Cladosporium sphaerospermum, Weignella dermatitidis, and Cryptococcus neoformans. These organisms actively seek out and colonize zones where radiation levels exceed the normal planetary background by a factor of 500. All three share a distinct physical trait. They are heavily pigmented with melanin. Traditional biology views melanin as a passive shield, a microscopic umbrella that blocks harmful ultraviolet light. But if melanin is strictly defensive, a structural problem emerges. Shielded organisms should merely tolerate a lethal radiation flux. Yet these fungi actually grow faster when exposed to it. Research in PLOS ONE reveals how this works. When high-energy gamma rays hit the fungal cell wall, they trigger a distinct physical metamorphosis. It starts with Compton scattering. High-energy photons violently collide with dense atoms in the cell wall, fragmenting into weaker secondary waves. Inside, melanin acts as a net. Its stable free radicals intercept these photons, capturing their energy and stepping it down into manageable amounts. Scientists measured this effect using electron spin resonance spectroscopy, tracking changes in magnetic fields. The data proved radiation directly alters the electronic structure of melanin. The radiation doesn't just hit the fungus, it changes the physical state of the cell's outer layer, allowing the organism to process high-energy photons. This altered electronic structure transforms melanin from a passive shield into an active biological battery. This chart maps the melanin's capacity for NADH reduction, its ability to transfer electrons. Non-irradiated melanin sits at a low baseline, but the irradiated samples show a precise four-fold spike in electron transfer. To find out exactly where this transfer was happening, researchers ran cellular dye tests called XTT and MTT assays. The results confirmed that this concentrated flow of electrons occurs entirely outside the cell proper, right within the melanin-rich cell wall. By capturing and converting energy extracellularly, the fungi bypass the complex intracellular machinery required for photosynthesis. Melanin functions as a biological transducer, taking the energy of a radioactive environment and converting it directly into metabolic fuel. To test the absolute limits of this biological conversion, scientists designed a starvation experiment. They measured survival using colony-forming units, or CFUs, which count how many viable fungal cells are actively reproducing. They took normal, melanized fungi and albino mutant strains that lack melanin entirely and placed both in an environment completely devoid of carbon-based nutrients. Over 30 hours, the albino mutants flatline and starve, but the irradiated, melanized fungi trace a steep upward curve, yielding 2.5 times more living colonies. Transitioning to a volume comparison reveals their metabolic footprint. The melanized fungi incorporated 3 times the amount of carbon-14 acetate, proving intense biological activity despite lacking food. Under absolute starvation conditions, these organisms manufactured life out of radiation and empty space. This localized laboratory anomaly scales up to the paleobiological history of Earth. The fossil record contains heavy concentrations of melanized fungal spores dating back to the early Cretaceous period. Radiotrophy likely allowed these organisms to survive historic magnetic field collapses and massive spikes in cosmic radiation. That ancient survival trait is exactly why dense, melanized ecosystems are so rapidly reclaiming the exclusion zone at Chernobyl today. This biological blueprint offers a pragmatic framework. Harnessing radiotrophic melanin could provide sovereign energy capture for deep space travel and engineer a new method for the bioremediation of nuclear disaster zones. The most hostile environments we have created have instead revealed how biology can adapt to utilize energy sources we previously considered purely destructive. Dive deeper into the hidden secrets that shape our reality. Click the subscribe button, activate the bell, and see you in the next scientific investigation, only on TEMBODocs.